This Week in Virology, the podcast about viruses, the kind that make you sick. Hello, Harvard Virology! Wow, I say jump, you say how high, right? That's great. From Microbe TV, this is TWiV, This Week in Virology, episode 1253, recorded on September 12th, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. 1253, that's my birthday. January 2nd, 1953. We are in Waterville Valley, New Hampshire for the Harvard Virology Retreat. And I was last here at the retreat, not in this location, but at the Harvard Virology Retreat in 2011. And one of my guests in the audience, David Knipe, and Phil Kranzush was another. That was fun. Now I'm back, and thank you for having us back. I guess you like TWiV, right? This may be the last one, though. My guest today from Harvard University, Aaron Schmidt. Welcome.
Thank you, Vincent, and thank you very much for accepting our invitation to come here to be our keynote speaker for our retreat.
I drove up with Aaron, so he's obviously an okay driver, right? Also joining us in the middle, a GS5 or G5. Is it G5 or GS? G. G is the government level thing. Yeah, you're right. G5, Tatum Sass. Welcome to TWiV.
Thanks so much for having me.
And at the end there, Ben Gewurz. Welcome to TWiV. You got a mic?
Yeah, thanks. Yes, I've watched a lot of TWiVs. It's amazing to be on this side.
Good. Well, we're going to put you through the paces. So, not only did you pick TWiV as the keynote, which I think is very good, right? It's different. And that's what I always wanted to do with these podcasts, do something different. But you picked the panelists, too. I had no say in this, so thank you very much. And if they do well, it's your doing, and if they don't, it's your doing. So by the way, I have stickers here, right? Because I want you to advertise for Microbe TV. You can stick these and come and get them afterwards, okay? Don't forget. I don't want to take them back with me. You know that I did a postdoc with David Baltimore, and he just passed away September 6th, not too long ago. David Knipe was also a postdoc. Student. Wow, you look so young. We didn't overlap, but I used to see you at parties and so forth. We got to be friends, and so it was a big loss. David was a fantastic scientist, as you know, and I assumed he would live forever because you could always call him or email him, and he'd answer. So it's too bad because he was amazing, and I thought he was the best scientist I ever knew, frankly, the brightest biologist in the world. So it's too bad. But I had interviewed him for TWiV 100, which, oh my gosh, that was 2010, ages ago. And Monday, the day after he passed, I listened to the whole thing, and I said, oh my gosh, this is actually good. The sound was good, and he was great, of course, and we asked good questions. So I reissued it, and when I went to the archive to look for the audio files, I found a video that I had never released. I put the camera, I was a novice back then, I put the camera on a tripod and I just pointed it at him because I didn't know how to get me in it, I didn't know how to get the two people online in it. I said, and I never released it, but it's there now if you want to see him answering the questions. And you know, I'm not a great video editor, but at the end, I thank him and he says, thank you, and then I just have him fade away and the chair is empty. No, I'm not Quentin Tarantino, but I just thought it was cool. So go check that out. It's over at our YouTube channel, microbe.tv. If you enjoy these programs, we'd love to have your support to do them. So we bring you science, real science, no lies, no disinformation. We give you the science as it's done by scientists. And if you like that and you need to know this, we'd love your support. We don't do ads on our programs. We depend on listener support. Microbe.tv/contribute. Okay, Aaron, you're the head of the virology program, yes? So, what is a retreat?
A retreat is a formal gathering of it. No. It's one of the highlights, in my opinion, of the entire program, and it's a chance for all of our students and some of our faculty to gather together to celebrate the science that everybody is doing, and also an opportunity for all of us to get together and join together to discuss science and celebrate each other. And so I think it's incredibly important, especially in the current climate, that there's an opportunity for us to gather and again, to celebrate each other.
So this is something you do every year, right?
Yes. So this happens every year in the fall, and it's a chance to welcome our incoming first-year graduate students who have been accepted into our program and are about to begin their very long, arduous, long PhDs. And this is before they've had a chance to select who they're going to do their thesis research with. So it's an opportunity for our incoming class to be able to interact with other class upper years and to hear about their experiences that they've had with their mentors and their thesis advisors, and give them an opportunity to learn about all the science, the great science that is being done in the virology space at Harvard. And so it happens every single year in the fall, and it varies from where we go, whether it's in the mountains here, where everybody loves to hike apparently, not me. But then it's also an opportunity that we can go to Cape Cod, which is my favorite place to go to, Provincetown in particular, which David notoriously hates. So when I was a student, it was not an opportunity for us to go there because David did not like it. But now that I'm head of the program, we will be going to Provincetown again, even though we're not there this year.
Yeah.
Thank you, Suzanne.
Where are the G1s? Raise your hand. I want to see you. Oh, cool. Did you have a good retreat? All right. Well, you're not going to say no, right? Okay. So these are great. I had a great time at the last one, and I'm really glad to be back. This one's even better, actually. So one of the cool things at this retreat is last night we had a trivia contest, right? Which is really good. Every table was a team, and we got really great questions. And like there was a whole series of clips from science fiction movies, right? And you had to guess what the movie was. I thought that was cool because Ian Lipkin was the script consultant for Contagion. And so he actually gives a talk where he goes through all the virus-related movies and critiques them. And the earliest one is in the 1920s with Vincent Price. It's a black and white issue. And I think it was reissued later. It was redone in color with some other actors. But you should check out that. If he's ever around and he's giving that lecture, you should check it out. So let's see. There was a TWiV set of questions, which I thought was really cool. I got all those right. But there was one that was kind of confusing. What was the latest TWiV? And there were 2 this week, right? So you had to modify that. And then, so the bit about Contagion made me think. So you had played a little clip, and was it? I forgot the actress who was talking. She says it's a hybrid between a bat and a pig virus, right? Something like that. Yes. And then it turns out to be negative stranded. So what's wrong with that scenario? Does anyone know?
Negative strand viruses don't recombine.
Negative strand viruses don't recombine. That's right. So Ian screwed up. I hope he doesn't watch this. It's okay, Ian. It's like someone told me, who cares, right? Except us. So that's really cool. And here's another thing that I picked up during this meeting. How to pronounce words. This is from you.
Oh no.
So how do you pronounce the word HEPES?
Oh gosh. Okay. I've always said heaps. However, the great David Knipe corrected me and said it's heepies. So from now on, I fear I'm going to have to switch.
It sounds like herpes.
What do you thought?
Oh, it is 100% heaps,
Heaps.
without question.
What about you, Ben? Ben?
I'm going with heepies because it sounds like herpes.
So David and I are the same era, we used to call it hippies, right?
Hippies, always.
But it doesn't matter. I mean, it's just an abbreviation for a chemical, so it doesn't really matter, right? All right, here's another one. This one is from you guys, C-O-O-P. How do you pronounce that? C-O-O-P is a store in your place, right, in your campus. How do you pronounce C-O-O-P?
It's the coop.
The coop?
Coop.
Coop. So, Cornellian, where's the Cornellian here? How did we call it in Cornell? Do you remember? C-O... The co-op, exactly. And I didn't even prompt her. Look, we called it the co-op, right? And there was no hyphen either, so it just varies. And one more. This is my favorite. Programmed cell death. How do you say that? It's the A word, right? A-P-O-P-T. Do you know programmed cell death?
No, I'm just an ignorant virologist.
How do you pronounce it?
Autophagy?
It's okay. You're a nice guy. Do you know what programmed cell death is?
Oh, I'm going to say it wrong. I've always said apoptosis.
Right. Yeah, that's how I say it, but I used to say it. What about you, Ben?
I also say it that way, but I've heard Horvitz say apoptosis.
Apoptosis, yeah.
Mm-hmm.
Who says apoptosis? I used to say it all the time, and then I co-authored this virology textbook, and they excoriated me for saying apoptosis. They said, "You're so unsophisticated to say that." So I got shamed into saying apoptosis, but now I have the whole room behind me now, apoptosis, because it's a helicopter, right? And I think if you go to the original paper where the guy used that word for programmed cell death, he tells you how to pronounce it, and I think it's the hard P. Anyway, I think it's fun we always do this on TWiV. We'll pronounce a word, and then Kathy Spindler will say, that's not the way you pronounce it, or that's wrong. I think that's a lot of fun. It illustrates, remember, this is programmed for non-scientists as well as scientists, and it just illustrates how flimsy science is. Okay. Anyway, those are my memories from retreat. So thank you again for inviting me. But now we focus on you guys up here, and I want to start with your histories. I want to know from the very beginning. Oh, this is something that people don't get. When you were haploid, do you know what that means?
No.
Really?
No. How far back do you want me to go?
When you were haploid.
When I was a haploid? All right. So what is a haploid again?
So you had two parents, right?
Okay.
And their gametes, they're haploid, and they come together. So I want to know where the parents lived.
Okay. All right. I can do that. Just want to make sure I was on the same page as you. So my parents were born and raised in Milwaukee, Wisconsin. So that's where I am originally from, and I was born and raised there in the Midwest. And my mother worked for a law firm, and that was her education. And then my father was a computer engineer, and I then became a product of them. And
you became diploid.
I became diploid, yes. Not diplo, but diploid. Okay, nobody got that. All right, that's cool. Good. I'm batting 1,000 today. So I went to high school in the Midwest, and as I said, was raised entirely there. And then I love to tell this story because it kind of makes the trajectory of how I became a scientist. And so I went to a very strong theater magnet school when I was in high school, and I loved both theater and science, trying to keep those two going simultaneously. And for theater, had done and toured 2 shows overseas as well while I was in high school. I was John Proctor in The Crucible, and that was one of my favorite roles. I'm happy to do a monologue right now if you would like. But the point of my story is that when
remember it?
I do.
Do a few sentences.
This is a stand-up moment.
Absolutely not. Maybe at the end on the director's cut that we can do later. And so the point of my story is that when it came to deciding where to go for college, my parents actually wanted me and encouraged me to go to conservatory and to go into theater and to go into dance. And that is something where most parents don't encourage that. And I actually wanted, and was trying to convince them that I wanted to go into science. And so I actually had the argument that I wanted to be a scientist, and that if I wanted to, I could eventually go back to theater and dance, but that I would first start with science. And so I won that argument, maybe. And then I went to Emmanuel College for my undergraduate degree, which is in the Longwood Medical area, just very close to Harvard Medical School. Fun fact, I was the first co-ed class of Emmanuel College. So it was an all-women's college for many, many years. And then there was a decision to go co-ed. And so I was the first co-ed class. And some may ask how long do you want me to ramble on for? Just keep going? Okay. Some ask how I actually ended up at Emmanuel from Wisconsin, and it was because of my physics teacher in high school. She went there for her undergraduate degree before she went on to Harvard for her higher education. So she wrote a letter to the admissions committee at Emmanuel College because they were asking for people with Y chromosomes. And so she encouraged them to reach out to me. And so I went and came and visited, and then the rest is history. I went there for my undergraduate, got chemistry, biology degrees, and then I applied directly to graduate school. And Harvard Virology was one of the only biology-focused programs that I applied to. I was going to go into chemistry instead of biology. And it was during my undergraduate honors thesis in biology that I met Todd Allen, who Tatum was advised by during my undergraduate, and wrote my senior thesis with him. And he was the one that encouraged me to apply to the Harvard Virology program. And for better or for worse, David Knipe accepted my application and invited me to come as one of the incoming class back in 2005. So I'm a G20 at this point. I guess that adds up. And so that's where I did my PhD in virology, and I did it with Steve Harrison, who is a friend and of David Baltimore's. And I did my PhD with Steve, and then I stayed on as a postdoc as well. And then in 2018 began my independent lab in the microbiology department at Harvard and at the Reagan Institute of Mass General, MIT, and Harvard. Yes.
So the science, you can hear me in the back, right? The science came from you. It didn't come from your parents, the idea of going into science.
No, there was nothing that from that pedigree, if you will, encouraged me to go into science, especially the biology or chemistry.
Okay. All right, well, we're going to talk about your science. No, you don't have to give it to her, it's okay. I'm not picking on you, don't worry.
It's all right.
It comes later.
We're going to talk about your science. Okay. What about you, Tatum? What's your, when you were haploid, where was that?
I was born and raised in Seattle, Washington. I did a short stint in New Jersey, moved back to Washington.
Didn't like New Jersey, huh?
No, it's just, my dad's in sales and marketing for automotive industry, and so we really just moved wherever his job took us. And so while I went through elementary and middle school in Seattle, I then moved to Dallas, Texas for high school. And so I kind of went slowly making my way to the East Coast, then over to Clemson University for undergrad. I got dual degrees in biochemistry and genetics with a minor in business administration because I thought, oh, hey, you know, maybe if I either have my own lab or go into industry, it'd be good to know how to balance a, spreadsheet. And then eventually made my way up here, and it's been an honor ever since.
So you wanted to be a scientist early on as well. There was no parental influence.
No. I had an incredible 6th grade biology teacher, Judy Barrere, and she had kind of pulled me aside and said, you don't seem very interested in what we're learning right now, which was what I felt at the time was very male-centric of dinosaurs and space. And so she pulled me into a special group and taught me about vacuums and that air had weight and chemical reactions of cooking. And that kind of inspired me to go into science as a whole. And then in college, my sophomore year, I got really sick. I was taken to the hospital with an 105-degree fever. No one could figure out what was wrong with me, and they ended up doing a full viral panel. It was adenovirus, something that typically doesn't make people sick. And so when I went through that, I started to think, what's different about me? Why are my cells reacting so differently than the majority of the population? And that's what kind of spurred my introduction to virology.
Cool. All right. Well, we're going to hear about your work as well. And that brings us to you, Ben.
All right, so I have to be the one in the group who was kind of pre-programmed in the haploid stage. So my father grew up in Indiana, my mother in upstate New York. They met in a lab. So my father was a postdoc at the time, University of Minnesota in Bob Good's lab. And so my mother was a visiting medical student there. But I will say I have two siblings who stayed far away, as far away as they could from this stage and from research. One went into law, one into government. So I'm the one who stayed on the pre-programmed path.
All right, so tell us where you went to school.
So went undergraduate to Stanford, then came here, I won't say the year, but came here for an MD, PhD, and have not left ever since.
Right,
so did my PhD with Hida Pluch, who was at the time the head of the immunology program, studying how cytomegalovirus evades the immune system. He actually was very persuasive and talked me into doing something I never would have guessed I was going to do, which was structural biology, studying how does this virus evade the antigen presentation pathway. So we've talked a lot about David Baltimore, but would also give a shout out to Don Wiley. I wound up in Don Wiley's lab at the time. It was a joint lab with Steve Harrison's lab. It was a really special place to be. It was kind of fun to be doing structures related to the MHC in that environment, because I don't know if all the students know that that's where the first MHC class 1 and 2 structures were done. You had an interesting TWiV at the American Society of Virology meeting where Pamela Bjorkman told the stories behind that.
Yeah.
It was interesting to hear what they thought they were going to find at the time. So from there, stayed in the herpes virus family. Still in the haploid stage here, scientifically, and did my postdoc with Elliot Keefe, who was a prior, before David Knipe, he was the prior head of the virology program. So studied Epstein-Barr virus pathogenesis, which I guess we'll come back to.
When I came to Boston, it was amazed at how many good scientists that were here, right? Just you've rolled off a bunch of them, but so many more, and more than I'd ever seen anywhere else. Especially virology. Virology was really, really strong. It was a great place to train. So this haploid thing, I used to use it all the time, and then once I asked some distinguished scientist, and she looked me straight in the eye and said, "What?" She didn't get it, and I said, "Well, I guess it's not obvious, right? So I don't use it anymore." But I think it's cute, right? So I thought I'd try it on you, so I'm going to revive it, because you reacted well. Let's talk about some science. I want you to give us, Aaron, an overview. You can't sleep up here.
I'm not sleeping. I'm worried about what's going to come out of your mouth next. That's all.
I'm not gonna. It's your science. You know it better than anyone. Certainly better than me. I just want you to start by giving us an overview of what goes on in your lab. Sure. What you know goes on.
Yeah.
Well, I don't know exactly what goes on, but I have a few graduate students, so if Giancarlo or Lamont or JC want to weigh in, they can. No, but in general, my research program has largely been centered on using protein engineering as ways to tinker with proteins in order to kind of direct or redirect the immune response to understand how we respond to pathogens, in particular viruses. That's what we focus on. And so what I'm very interested in trying to understand is what is the way that our immune system sees a pathogen and how antibodies are made to it, and how can we use that information to potentially guide next generation viral vaccines. And one thing that we're interested in trying to understand is once you make an immune response to a particular pathogen, how does that pathogen sense that immune pressure, and how does it evolve away from that immune pressure, and how does viral escape happen, and how does viral evolution happen? So we're trying to use that information to ultimately try to design better vaccines or new vaccines against some of the most pressing viral diseases around.
So as you know, the head of HHS doesn't believe in vaccines, right? He doesn't believe in infectious diseases, in fact. So do you think there is a future for new vaccines?
I just said I was worried about what was coming out of your mouth.
I know it's a hard question.
But it's an important one. And then, all joking aside, I absolutely believe that there is a path and a necessary path for new vaccines to be developed. I'm a strong believer in vaccines. I'm an advocate for vaccines. And I think that the work that we're doing can certainly be used as blueprints for approaching potentially new pandemics that could enter into the human population. And so I would vehemently disagree with what is currently being put out by our government, and am a strong believer in vaccines and the importance of making sure that our communities are well-versed and knowledgeable in what vaccines can do in terms of protecting them against viruses and other diseases that could have other rather severe consequences.
I'm always amazed. Great audience, really good, thank you. I love it. I feel like I'm Johnny Carson. I see so much cool technology that can be applied to vaccines, and it makes me sad in a way that we have a leader who doesn't embrace that, right? But I agree with you. I have great optimism, and we're going to get past this. We will. We have to, because the alternative is much too much suffering. All right, so I read a couple of your papers, and it was good to put me asleep. Just kidding. You're good to joke on. He's a good jokester, right? Oh, you know this already, right? Oh, okay, I didn't know, because I'd never met him before. So here's one. Conserved sites on flu H1H3 HA recognized by human antibodies, right? These can be used as a template to design a vaccine that elicits them. So I always wonder how you could do that. So you know which antibodies are important, which epitopes induce them. How do you focus a vaccine on those?
Yeah, so that is a great question. And so what we often do is look at how, in that particular paper that you are referring to, is actually, the lead author on that was Daniel Maurer, who was actually a virology graduate student who was recently graduated and is now at Moderna, doing research there as a scientist. And so one thing that we're interested in doing is, as you pointed out in that paper, is that we identified human antibodies that recognize particular parts on the influenza hemagglutinin in particular. And the hemagglutinin and the vaccines that we have against flu are largely, from year to year, varies quite a lot, but in terms of efficacy, and it's no greater than usually about 50 to 60% efficacious from year to year, and then sometimes it can be as low as 3%, and sometimes not effective at all. And so what we're trying to do is to understand from those those vaccines that are used routinely, the seasonal vaccines, or through natural infection, the antibodies that are made in response to those, either natural infection or the vaccines. And what we did in that paper was to understand where these antibodies bound on the hemagglutinin, use structural biology to determine their epitopes. And once we understood where these antibodies were binding, we then further characterized these antibodies and what we were interested in terms of how broadly could they bind to other different hemagglutinins, or how potent were they in neutralizing the virus. And we use those as metrics, as ways to say, these are the types of antibodies that we want to potentially elicit by next generation viral vaccines, because they have quote unquote superior qualities than the other antibodies that are made by our standard seasonal vaccines or through natural infection. So we're trying to fish out those needles in the haystack, if you will, that are the best, baddest, broadly neutralizing antibody, which I don't often want to go after, but at least is something that can be used then for a template, because once we recognize on the structural level how this antibody sees the hemagglutinin or the epitope of interest, we then take that epitope and then design strategies for our immune system to only see that epitope. And so we use that, one of the approaches that we take is epitope scaffolding. So we take that particular epitope and put it onto something that our immune system has never seen before. And so that the focus can be just on that epitope. Or an orthogonal strategy that we use is to take that epitope that is naturally on the hemagglutinin and use glycans or sugars as a way to sterically block the ability of the immune system to see the other parts of the hemagglutinin that would otherwise distract from that particular epitope that we're interested in. So we use a lot of, as I said in the very beginning of this, protein engineering as a way to tinker with these proteins, as a way to trick the immune system to only see the epitopes that we're interested in getting our immune system to recognize.
It's interesting you say 50% efficacy, right? So Daniel Griffin always says, he has a patient who is hospitalized with influenza, and they say, hey, doc, I got the flu vaccine and I still got sick. And Daniel will say, but did you die? So you may get sick, but it often protects you. So you would like broadly neutralizing epitopes because that way you might not need a vaccine every year, okay? So, and there are two places where, there's one main place where you find such broadly neutralizing antibodies, right? Mm-hmm. Which is the stem. Tell us about that.
So you hit a hot button issue on me for this one.
Yeah,
No.
Go for it.
In all seriousness, I think that for me personally, I do believe that the stem can be a good target for antibodies that could help contribute to control and protection against influenza. However, I am a huge proponent of the receptor binding site and the head domain. And so I sit on the opposite camp than Peter Palese and others, and Florian Kramer, Daniel Lingwood, my esteemed colleague at the Reagan Institute, who also believes that the stem should be the target of next generation influenza vaccines. I have no doubt that they will help as a useful component, but those antibodies are largely important for ADCC or ADCP dependent mechanisms of protection. And so those are only after the virus has actually established an infection. And so the first line of defense, in my view, should be those that block the receptor binding site and actually inhibit the virus from actually getting into the target cell before it can establish an infection. And so our efforts have largely been focused on finding ways to direct our immune system to that conserved receptor binding site, because the vast majority of influenzas, except for several of the bat H17s and H18s, use sialic acid as a way to get into a target cell. And that receptor binding site that chelates the sialic acid in order to enter is largely conserved across many different influenza hemagglutinins. So that's the target that we should be going after, in my view, and secondarily should also focus on the stem region.
So the stem antibodies are not neutralizing, you're saying?
No, they in the traditional assays that are used to determine neutralization, you do not see neutralization with stem-directed antibodies, only with those that bind the receptor binding site.
That's some nice discussion here. So there are conserved epitopes on the head.
Yes.
You're saying, are those inducing neutralizing antibodies?
That is correct. Yes, and one thing that I also want to. I don't necessarily subscribe to the one epitope only in terms of receptor binding site or stem. I think we need to think of the hemagglutinin as a continuous epitope, that there have been antibodies that can target various portions of the stem, or excuse me, of the entire hemagglutinin and not just one discrete epitope, and that all of these working together could help control the spread of these viruses or influenza.
So the conserved head epitopes, are they also broadly neutralizing?
So those that target the receptor binding site, what you often trade for potency is breadth. And so those antibodies that are very good at binding the receptor binding site for H1 influenzas, for example, they are broad against many of the H1s, but then can't cross, if you will, to group 2 influenzas like H3s. However, they are extremely potent within the particular subtype of interest. And so I could imagine that if you're trying to develop a next generation viral vaccine for influenza like we are, that it's a quite tall order to suggest that you would get one that is universal against all of the influenza subtypes. And maybe a more practical way of approaching it is to have a very good next generation vaccine that targets group 1 influenzas and one that targets group 2, and to use them together as a way to combat both group 1 and group 2 influenzas, as opposed to trying to get one that covers all of them.
All right, and what you said before is you would make a platform to present these epitopes, which is not related to influenza virus at all, right?
That's correct, yes.
Okay. So the best we can do is have probably 2 groups of broadly neutralizing epitopes that generate antibodies, right? And can you get resistance to those broadly neutralizing antibodies?
You can, and so there was some work that we and others have done that when you take these very potent H1 specific antibodies that block the receptor binding site, you can, for that particular antibody, select for resistance and show that even though they are very potent, that resistance can develop quite rapidly, actually. Even after 1 or 2 passaging, you can get resistance mutations that come up. However, the key observation in those studies is that, yes, you can lose binding to one of the antibodies that recognizes the receptor binding site, but through structural analyses, you can show that there are other antibodies that still recognize the receptor binding site and the conserved elements of that, but that approach from a completely different angle. And because of that, they are not sensitive to that mutation that escaped the first set of RBS-directed antibodies. And so the idea there would be if you had a immunogen that could elicit antibodies that target from a variety of different angles, all recognizing that conserved site, that the likelihood that you would get resistance to the entire class of antibodies that are recognizing that would be very small. So even though you can get resistance to one, I don't think that you would be able to get resistance to all of them, because at that point you'd be compromising the viral fitness and the ability of that virus to chelate or bind sialic acid to get in.
So your message to the non-scientists listening is that we're going to have a different kind of flu vaccine in the future.
I think the way it was designed, yes, but in the way that it's going to be administered is going to be the same.
Oh, sure.
If that makes sense.
Yeah.
I just want to make sure that that's clear, that this isn't going to be a new platform or modality, I would say.
I just can't help but mentioning there's this NIH initiative to make next generation flu vaccines, which is just to take virus and inactivate it, just like we've always done, right? And they're saying this is brand new technology that's going to protect against all phyla. Do you remember? Did you hear that? All phyla. What do you think about that? All phyla.
Do I have to answer every question you ask me?
It's okay.
No, I agree with your incredulousness at that statement.
Well, phyla. Okay. One more paper, then we'll move on. This other paper caught my eye. Antigenic drift expands influenza viral escape pathways through recalled humoral immunity. Okay? So recalled, is that memory?
Yes.
Why don't you call it memory? Is this an immunological thing that you have to have different words, or?
I'm going to punt on that one and say the editor made us change the title
really?
of that.
Really? Because originally antigenic sin is basically memory, right? And it's just special for flu, but
Sure.
it's memory, right? So what does the title mean? That's really hard for a non-scientist to figure out.
Can you read me the title again?
Antigenic drift expands influenza viral escape pathways through recalled humoral immunity.
So that, again, is from Daniel Maurer. So maybe he should be sitting here fielding these questions instead of me. So what that essentially meant is that as the virus evolves in the human population and drifts, so when H1s, for example, were reintroduced into the human population in 1977, and as that virus circulated in the human population, we made immune responses to that virus as in your lifetime, whether you were exposed through vaccination or if you were infected with that virus. And you essentially recalled every time that you were infected with that virus a recall memory response, so those antibodies that recognized features of that virus that was conserved from year to year to year, even as the virus was drifting. And so what the paper was looking at is how does that recall response actually influence both the evolution of the virus, but then also are there many antibodies that by targeting the influenza hemagglutinin could potentially, I've even lost my train of thought. Let me start over there. So the point would be that as the virus is drifting and it's recalling that memory response, that the virus is finding many more opportunities to escape that humoral response. And so it's actually doing a disservice to have that memory recall, and that what we'd want to do is actually to stimulate new responses that aren't relying on the memory recall from those previous exposures.
I just remembered who he reminds me of. Terry Dermody.
Who's Terry Dermody? Oh.
You don't know who he is?
I know who Terry Dermody is.
Okay, look him up. You got a similar happy expression. Okay, so if you first see an H1 influenza virus, and then as long as it drifts, you're going to get original sin, right?
Yes.
Or what's the better word? Original antigen.
Antigenic sin.
What's the original antigenic sin is too religious. What's the
Imprinting?
other? Imprinting. Thank you. Imprinting. So H1 will never be different enough to not make a memory recall, right?
That's correct.
But will
Except,
an H2 be different
oh,
enough?
absolutely, yes. And so
Okay.
we observed that in 1977 all the way up into 2009, that H1N1 was very similar as it evolved in the human population. But in 2009, that was essentially a reset of H1s because that was the introduction of a truly antigenically distinct hemagglutinin that we had never seen before, or at least some people, the vast majority of the human population hadn't seen before. But what was curious about that is that once that was introduced into the human population in 2009, some of our older population had experienced an influenza that was similar to that H1N1 from 2009 in 1917 when the pandemic happened for the H1. And the recall response that you got from that was actually targeting the conserved stem region, going all the way back to the original, because that was the only thing that was conserved between that original H1N1 in 1917 and then all the way to 2009.
Okay.
But a reset with a new subtype of influenza will definitely cause a non-recall response and a de novo one.
Okay. All right. I could ask you a million questions, but we have to move on because we don't have so much time. Okay. You're off the hook for now, but I'm going to come back to you. Okay. Tatum, tell us what goes on in your lab. Are you still working or you're finished?
Oh gosh. As you always say, you're always a student. You're always learning.
Yeah,
and,
but doing experiments.
Oh yeah, no, I'm hoping to wrap up most of my wet lab work by about December, January, and then, knock on wood, hopefully start writing my dissertation around.
Mm-hmm. Whose lab are you in?
Yes, so I was formerly in Todd Allen's lab. He passed away about a year ago, very tragically and unexpectedly, but I've had such incredible support from the Harvard Virology program, the Reagan Institute, and the colleagues in this room that have allowed me to continue on his work and his research, and I couldn't think of a better way to honor his memory.
What was the general goal of his lab work?
Yes, so Todd's lab had previously been focused on, it's always been pretty HIV, HCV-centric lab. I'd say the past 10 years, the focus was developing CAR T cells for HIV. And for those of you who don't know what a CAR T cell is, it's a chimeric antigen receptor, which targets in an HLA-independent manner. So you're kind of bypassing your natural targeting system on T cells.
So the idea would be this is going to kill HIV-infected cells. Is that correct?
So as the virus goes through its life cycle, the envelope is presented on the surface of an infected cell. And so our hope would be to target those infected cells.
Of course, the cells that are latently infected are not going to be targeted because they don't have anything on their surface, right?
So there's conflicting evidence in the field. Some people would say that there are small amounts of envelope that are expressed on latent cells, and so it'd be a bit more difficult to target because the numbers would be smaller. But I think there's a possibility that those could be used too, or in combination with like a latency reversal agent.
So what did you work on? And you said you continued that work, right? So what was that?
So my goal has been to improve the targeting domain or the ectodomain of these CAR T cells. And so I've been trying to modulate the CD4 component, which is what we use to essentially recognize HIV envelope, in a couple different ways.
So the basic idea was that normally a CAR T cell molecule will have an antibody to a cancer-specific antigen, but you're using CD4 to target it to HIV, so cells that have the viral glycoprotein on the surface, right?
Yes.
And you gave a talk about that today, how you're optimizing. And was the bottom line, the higher affinity, the better killing you get?
As of today, yes. I would say that there's definitely a range. You don't want to go too high or you won't get re-engagement, but with the affinity that I've been able to enhance and achieve, it seems like that has been significantly better at killing these cells.
So what would you call yourself, a virologist or an immunologist?
Oh gosh, I think I lean more towards a viral immunologist. My politically correct answer for this room, but.
So what result over the years got you most excited?
Okay, I I think this is a great story just about scientific perseverance. My first year in the lab, I wasn't able to get any sort of CAR to express on a cell surface. And I had to go back and I looked at the original design, and it turns out I had a 70 base pair deletion in my gene block when I had done the original cloning for this plasmid. And so when I found that deletion and then recloned and was able to go back and express a CAR on the cell surface, I feel like my project went from a year of troubleshooting to immediate results. And so, you know, perseverance in science is a skill that you acquire over time. And I feel like I had a tough go of it, but I think ever since then, again, knock on wood, because I'm still doing wet lab work, things have continued to go uphill from there.
What do you love about science?
Oh, I do something different every day. I go into lab and I'm excited to be there because I know I'll have a new result or a new assay, or I think just something different, something new. There's, as you say, there's always something to learn. And so I think just going in and doing something different, I mean, that's what gets me excited.
What don't you love about science?
Oh gosh. I think something that frustrates me, again, I come from a very non-scientific background, is how jargon heavy science is in general. And so when I'm explaining things to my family or friends or people outside of the field, it can get a little difficult to try and come up with the right words. And I think just in general, communication is really important to be able to present your work in a way that anyone can understand. And so I think something that frustrates me about science is how scientifically oriented our language is.
Do you think that is something you could help fix in the future?
I sure hope so. I feel like I also sometimes struggle and will use words that maybe are difficult for others to understand. You know, I'll say assay instead of I'm doing an experiment or a test, and people don't always know what that means, but I'm going to work on it, and I hope other people in this room are inspired to do the same.
Yeah, that brings back a David Baltimore memory. A student was saying, I did a blot, and he said, that's jargon, right? I did a blot. You think your parents would understand I did a blot? No.
Oh, absolutely not.
What are you going to do next, do you know?
Oh gosh, my hope is to go into industry. I think there are people that fall into two different categories in science, in my opinion. You're a big idea thinker, or you're a problem solver. And I feel like I tend more towards problem solving, and I think that skill set tends to lend itself better to industry.
Do you think industry can exist without academic labs?
Oh, absolutely not. I think that's where our incredible starting ideas come from. And then it's just kind of advancing them into a more accessible way for everyone.
Yeah, because there are some in this country who feel that industry should be doing everything, which is why I asked you that question, right?
Yeah, and I think that would be a big mistake.
I mean, industry just does not want to do a lot of the things that you're doing, for example. They will take your result and go with it, but they don't want to tinker. It's too expensive.
Step zero comes out of an academic lab where we have the freedom to experiment and do exciting things.
Are you worried about the future of science in the United States?
Yes. Something that many of my mentors have said to me recently is, are you even looking at jobs outside of the US? And to which I'll answer, I'm happy to go anywhere that will take me, but it's scary to see that, you know, the way our scientific field is trending right now is not the way I would hope and not the way I envisioned when I started in this program. And I'm so hopeful that we will get back on the right track.
Well, part of what we do is to send the science message to people, right? And people like you have to tell us what you're doing, and we tell it to them, and they have to appreciate how cool it is. And not only that, but makes them healthy, right? Okay, I'll come back to you as well. Ben, what would you say is the overall goal of your lab?
Yeah, so overall goal is kind of a tough one. To understand Epstein-Barr virus, how it works, how it causes disease, and how we can eventually stop that. I'm saying it's a tough one because it's a very tricky virus. There's a colleague, Lynn Enquist, who instead of calling it Epstein-Barr virus or EBV, he calls it everybody's virus or EBV because almost everybody in the world carries this virus. So it's learned how to get around detection, trick the immune system, even though it takes up residence right where you think it would not want to be, in one of the best cells of the immune system, to tell the rest of the immune system what it's carrying, what it's doing. And so it's an interesting problem to look at. And paired with that, that it causes very large amounts of diseases in very specific environments, but then the rest of the time, not at all. So in some parts, in some regions, very high incidences of a cancer that many people here may not even have heard of, called nasopharyngeal carcinoma, which will rival breast and lung cancer because it's such a common incidence in that region. Then you move to places that have year-round malaria called holoendemic malaria. Now it's, rather than that, a different cancer called Burkitt lymphoma that pops up, and that's where it was discovered. I was going to give a shout out that it was in part discovered by a graduate student. The Barr is Epstein-Barr, who was the first graduate student in the lab that found this virus. And a lot like what Tatum was saying, they both the PI and the student risked their careers on what they said was a wild goose chase that had to be chased at the time. But then you go to other situations with HIV co-infection. Now Hodgkin lymphoma becomes very common. So it interests me that in different situations, it causes different problems.
So many people are infected, right? Over 95%?
By the time, by adulthood. So
By adulthood.
Yeah.
Do they all get sick?
So most don't. So I think it's, probably the virus is trying not to cause disease so that it can live with us for our life. And that's how I think about it. It's trying to give itself this opportunity. I mean, another reminder of how good it is at evading immune systems that we don't recognize when we're shedding this virus. But there have been studies of probably grad students, I would bet, in the lab that was doing this. And just periodically, without you knowing it, the virus kind of awakens and starts to secrete itself. And so I think the virus is trying not to cause disease so that it can do that and make its way around the world.
But sometimes you do get ill from EBV,
Yeah.
like mono, right?
Yeah, so mono is probably, when, because of changes in sanitation, maybe other variables, it hasn't infected someone very early in life. So if it infects us when we're young, very young, 1 or 2, 3, you might, I have 3 kids who went through daycare. I'm sure they've gotten exposed to it, seeing kids put toys in their mouth, pass them around. They must have, but I could have never told when were they infected by all these viruses. So mono is typically when somebody reaches teenage years, 20s, and at that point gets exposed to EBV.
Do we understand how the virus causes mono? Like, what is it doing?
Yeah, no, I think not. And by the way, it's not just the virus. The virus is probably doing what it had done earlier in age hosts, but it's more our immune system that's causing, I think, a lot of the symptoms of the mono, and probably analogous to how you'd rather get chickenpox earlier in life than later, before the vaccine. Why that people would have these chickenpox parties to expose people to that early, because it causes a more severe response if you're exposed to it later. It's in the same family as chickenpox. So I think it's actually the immune system that's changing. But there is another theory that, there's several theories. It could be the amount of virus that you're exposed to that's different. So, another name for EBV is the kissing disease because it spreads through saliva. So when you pick it up later in life, you may get a higher inoculum than when you're a young child.
So once you're infected, the virus remains latent in B cells, right?
Right.
What reactivates it?
Right. So, although it is mostly B cells, it can get into other cell types, and you're kind of reminded of that because it can cause cancers in T cells, natural killer cells, rarely smooth muscles, weird cell types. So it can find its way to other places. So what reactivates? It's thought the best theory is so far is that it makes lifelong infection what's called the memory B cell. It knows this is a very long-lived cell that can live for decades, and so it's a good place to park itself until it's time to wake up. And so it's thought that when that memory cell is stimulated, so when that memory cell sees the antigen that it's specific for and starts to turn itself into a plasma cell, which is the kind of cell that cranks out antibodies and is kind of a factory for antibodies, it's a good time to leave if you're the virus, because that cell will stop growing. It might go to a kind of a place in the body near a surface that you might want to spread to. So there's something in that pathway that wakes up the virus and tells it this B cell is changing its character. It's time to get out.
Because those B cells are going to mature into something that's a dead end and eventually
it would
dies.
be because the plasma cell is
so
terminally
any
different.
infection could reactivate EBV, right?
That's right. Yeah.
And didn't we see a lot of that during COVID?
Yeah. So and there are some studies of people with long COVID where something like usually the number I see in these studies is about a quarter of people with long COVID have an elevated level of EBV DNA detectable,
Right.
and so.
And you would only look because they have long COVID, so you want to see what's going on, right? but people who don't have long COVID, who have just acute COVID,
Right.
they may also have EBV reactivation, but we don't look.
Yeah, they might. And then there's a number of people who get mono and then have this lingering fatigue afterward. That start,
Yes.
and some overlaps, and it starts to feel a little similar to what can happen with long COVID.
So in EBV reactivation typically is not symptomatic?
Yeah, typically not. So I think that's what I was trying to allude to before. That I would never know. I'm sure I've many times in life had this happen and have no idea when that's happening. So it's good.
So if we did big studies where we looked at people who were infected with influenza, RSV, SARS-CoV-2, if we looked for EBV in them, we would see a fraction
I think so. Yeah.
It's never been done, right?
I think that it's being done a bit, but I think not as much because it's sort of a passenger.
So what do you think that in long COVID it is an association or is it causation?
It's a tough one because it's in the B cells, and if the B cells are reacting to something, you can imagine.
Sure.
There's always a chicken and the egg question. Is it coming along? And it's in B cells. There's more reactivation happening because the B cell happens to be in is getting excited about something. So I think we need a little bit more information there. But it could be that because it's in a certain number of B cells that are getting activated, it gives the virus a chance to do its thing where it starts to make the 80 genes that have been hidden all along, and that causes a new problem.
So also EBV causes tumors, as you mentioned, different kinds, right? Not just B cell tumors, right?
Right, and I didn't mention before, but there's a lot of interest also in autoimmunity as well, and how exposure to EBV, maybe on that thought about getting exposed later in life might be a problem for people who are susceptible to autoimmune disease. And so if you have that sensitivity and you get EBV later, that might be a thing that puts you on the pathway to things.
Well, that's the MS connection as well. That's thought to be an autoimmune, right? Where you make an antibody to an EBV protein.
So in people with multiple sclerosis, you can see elevated antibodies to these, especially these Epstein-Barr nuclear antigens, some of the lytic cycle proteins. But also just the observation that it's very hard to find someone with MS that's in that group, that 5% who haven't gotten EBV. EBV almost always, if not always, precedes the diagnosis of multiple sclerosis. And so it's like there should be a few who don't have that. I mean, it's hard to find them.
Well, that's why this Army study was beautiful, or the military, where they took recruits who were EBV negative, and they take blood every year and see when they convert, and then they see when they get MS, right?
Right.
It's a perfect experiment.
Right, so they can look back in time.
Yeah, we did that paper on TWiV. You must have heard it.
Right? Yeah, that was interesting.
So back to cancers, how does it, so it causes B cell cancers. It must cause epithelial cells because nasopharyngeal carcinoma is
nasopharyngeal, and the other big one, by the numbers, there's more of these than the B cell cancers, is stomach cancer or gastric cancer. You're swallowing it all the time, and so it finds its way into the stomach.
And what cells are transformed?
So it's the gastric, the stomach cells. And so it may be that the cell is kind of abnormal and on its way to becoming cancerous, and the virus hops on board and makes it worse.
So do we understand transformation by EBV?
I think better on the B cell side, less. I'd say we understand it less well.
So is there an oncogene in the genome?
Yeah, so there are a number of oncogenes that the virus causes, and so, but it also can turn on expression of host oncogenes. So it's got two ways of causing a problem, either expressing its own oncogenes or turning on other ones that are very good at making things like B cells grow.
Mm-hmm. So any inflammatory condition which makes the cells divide might lead to tumors, right?
To that, and especially, I think the virus leaves the door open for I was saying before that it doesn't really want to cause disease, and so I think as it's activating the B cell and giving its program with its best punch, all of the transforming genes expressed. I mean, if there was no surveillance by immune T cells or natural killer cells, these would just immortalize in all of us and turn into lymphomas all the time. They're very visible to the immune system, and so I think the virus tries to give a chance to, allow the balance to be achieved.
Tumors are not of any use to viruses. They're accidents typically, right? And so for adenoviruses and polyomaviruses, we know that the tumor antigens makes the cell divide so that there's a DNA synthetic machinery, right? But what is it for EBV?
Yeah, so I think the best guess right now is that a little bit of virus gets in to start this whole infection. Rather than doing what some viruses do, which to me would be to make a whole bunch of copies of itself and spread that way, instead it will kind of lay low but make that cell that's carrying the viral genome replicate, and so it makes that B cell grow, and that might be what these are doing in part, but it wants to get to that memory cell. So another thing it's doing is activating these B cells so that it can push them into becoming these long-lived memory cells take it through that process that normally a B cell would do when it's reacting to some antigen, some immune stimulus.
Does integration of viral DNA play a role in transformation?
Yeah, usually not. So it's usually it's a double-stranded DNA episome or plasmid that's kind of it has a way of being attached to this kind of auxiliary. Once in a while it can integrate, but usually doesn't.
So you mentioned that there are these geographically restricted tumors, right? There's Burkitt's lymphoma in certain parts of Africa, and then we have nasopharyngeal carcinoma in other parts. So why is that? Do we have any clue?
Yeah, I think there's something environmental that's pushing it, and maybe something genetic as well. But in the case of Burkitt, you see that the combination of getting Epstein-Barr and having year-round or holoendemic malaria.
Malaria.
But we don't understand a lot about why that is. Is it you tend to see higher EBV viral loads when that happens? The malaria is a bit both immune suppressing and also stimulating the B cells.
Right.
So it's maybe a combination of things that allows the virus to.
Do people work on this, and are they trying to understand it?
Yeah, although not as much. When I look in the literature, not as much as I would have guessed. So it's hard to do because Epstein-Barr infects humans, not. It doesn't infect mice. So it's a little hard to come up with a system where you can actually put the two together, try to understand why the two is a recipe for that.
Yeah. Someone needs to make an animal model.
Yeah, for that. We're trying a little bit with an organoid system right now that might allow us
So are there therapies for these cancers, EBV cancers, that are specific for the presence of EBV, or are they just generalized cancer therapies?
Yeah, in general, I'd say it's generalized. There are some things in development where you have, instead of CAR Ts, we were hearing about Tatum, you can train T cells just to recognize the virally infected cells, and you can do this in a way where you have off-the-shelf versions that would match the particular HLA haplotype for whoever it is that you need to give them to. So there's ways to get those. This comes up especially after transplant, where because you've disrupted the balance if you've given someone an organ or a new bone marrow, you have to suppress a bit of the response to allow the that organ to be accepted.
There are viral antigens on the surface of tumor cells, right?
Yeah, a little bit. So there's some of these transforming proteins, a little bit of them are exposed, but much of it's happening inside the cell. But some could be presented on the MHCs.
Maybe you need to work on that, Tatum.
Yeah.
Not here, but somewhere else.
I'll try.
Okay. Do we need a vaccine for EBV?
Yeah, it's something that comes up a lot. I mean, the places that it would be helpful for would be, if somebody had escaped EBV and was at risk of having mono, it might help. If there was a vaccine that you could give to someone who, was getting into the range where they were more likely to have symptomatic mono, it might help them. So I think there's been some interest in that, including from the military. They have recruits that have that, who they don't want to have become disabled by mono. And then there's a connection. The same people who get symptomatic mono are at higher risk for Hodgkin lymphoma and also for multiple sclerosis. So I think those would be things that you'd wonder if a vaccine could steer someone away from.
Are people working on vaccines?
So there is interest in doing that, and it might not prevent someone from getting EBV, but it might just give enough of a boost that when kind of what we said before, when you made that comment of have you died from it yet?
Yeah.
It may not prevent the infection, but it might prevent the bad outcome.
Won't prevent infection because it's very hard to make sterilizing vaccines.
I think it's going to be a challenge. There's a virus that can reactivate at will throughout your life.
Yeah, so that's the problem with HIV, right?
Yeah.
You'd have to have a sterilizing vaccine to prevent the establishment of latency, and same with EBV. But it's not clear how that would impact disease, right?
Right.
That's the difficulty. Okay. All right, I have a question for all of you. A few questions. So with you, Aaron. So given the NIH cuts and the Harvard-specific cuts, how is morale?
I'm never coming on TWiV again, just like right now, even if you invite me. And you need to find your own way home tomorrow.
You're lying.
I am lying. No, this has been great. Honestly, I think that is why, going all the way back to the beginning of the importance of this retreat and why we're having it here now with our students in particular, is that given everything that is going on with our administration, with the NIH cuts, the attacks on science, the attack on viruses and the work that we're doing here, that many of our graduate students are actually actively working on, is to make sure that we as faculty are supporting our students and keeping morale up by having opportunities like this to come together and to actually celebrate science and celebrate the work that we're doing, because it is incredibly important. Because the last thing that I would ever want is for any of the people sitting in this audience to be turned away from doing science, because everything that they're doing right now is so critical to the next generation of preparing for pandemics, preparing for understanding how viruses cause cancer, the things that Ben has just been talking about, the things that Tatum has been talking about as well. And so it is our job to keep morale up, and so put a smile on your face and keep your head up and keep doing what you're doing, because it is incredibly important. And do not be dissuaded or discouraged by what is coming out of Washington.
Yeah, no, just to add to that a little bit, I guess morale is still good. People are excited about what they're working on. I think the postdocs are quite worried about people who've been training. So Tatum's talked about how long she's been at this, but the ones who then gone on to the next stage have kind of been preparing for what they're going to do next and wondering, will there be a job for them to apply to, and if there is, what will that be like for them? And so that's, I think, a place I've worried a lot about and tried to keep morale up a bit with.
Tatum.
Yeah, I mean, I'm not going to lie. There have been a couple tough moments. I mean, when you see your friends that have worked so hard on F-31 grant applications get them canceled, it's a little bit demoralizing. But I think as a community, we've done an incredible job of coming together and supporting each other. And I don't think I could ask for a better group of people to be doing this with.
So David Baltimore always used to say, only do science if you can't think of doing anything else. So I'm going to ask you, Aaron, if you hadn't been a scientist, what would you have done?
I would have been the TWiV podcast. I would have started my own. Honestly, though, no, it goes back to my story that I was telling in the beginning. I think that I would, in a heartbeat, have been an actor. I would have tried to do that. Whether I would have been successful or not, I don't know. But that's certainly something that I would have loved to have done, was to go into acting.
Tatum.
Yeah, my friends and I have always joked we should all open a bakery when, the science is not going so well. So, jokingly, a baker, but I really think I've always had an eye for math and science, and so maybe something a little more creative like architecture. I don't know.
So my joking one, someone in the audience here is wearing a shirt of the Herpetic Legion, which is this band that plays at the Herpes Virus Workshop. They have this persistence tour that reactivates once a year. So I think I'm going to join that as a keyboard player to fulfill that alternative fantasy of being a rock star. But so I actually do have another hat. So I did the MD PhD and I'm a physician scientist, but kind of surprised my interviewer for MD PhD program at Stanford when he said, if you had to pick one of the two, what would it be? I said the research, and he didn't believe me at the time. So I guess my other career would be the other hat, which is infectious disease.
Okay. All right, that is TWiV 1253. I can't forget one. 1253, 1253 from the Harvard Virology Retreat. I want to thank my guests today. Aaron Schmidt, thank you so much. Applause, please. You'd come back if I asked.
No.
Tatum Sass, thank you so much. And Ben Gewurz, thank you so much. I'm Vincent Racaniello. You can find me at microbe.tv. If you want to send questions or comments to TWiV, TWiV at microbe.tv. If you want to help us spread science, microbe.tv/contribute, and pick up some stickers and put them somewhere visible. And when people say, what is that? You can tell them this is science communication. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. Now, I want you to do the tagline. okay? This was one of the questions, I think, yesterday at the end, right? The kind that make you sick. So I'm going to lead you in, and then you're all going to say in unison, another TWiV is viral. Jesus, I forgot it myself. Okay, you've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Now I have to edit this. You say another TWiV is viral. Let's try it again. This is what we do at the end of the TWiV. You got it now?
Yes.
And you got to all say it together. I'm trying to make you part of this. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week.
Another TWiV is viral!
Oh, beautiful. Thank you.